Assalamu alaikum. We'll discuss structure of cell membrane today. But before we discuss structure of cell membrane, we need to just review the structure of phospholipids. Phospholipids, consist of uh, two substances, a glycerol molecule two fatty acids and a phosphate group. Okay, so a glycerol molecule, it has a hydrocarbon chain with three hydroxyl groups attached with them. To balance the valency of carbon, you can have two hydrogen here, two hydrogen here. So this is already balanced. So this is a glycerol molecule. Glycerol. Two fatty acids. Fatty acid, they have a long hydrocarbon chain which may be represented with an R and a carboxyl group attached with it. So if there are two fatty acids, each will have a long hydrocarbon chain which is represented with R here and a carboxyl group which is attached with this R group, and this is another fatty acid which has an R group with a carboxyl group attached with it, and a phosphate group which is represented or maybe written like this. Okay, condensation between glycerol and these two fatty acids and a phosphate group eliminates three water molecules. Okay, one water molecule will be eliminated here from this hydroxyl group from this fatty acid and hydrogen from here, and other water molecule will be removed from the other fatty acid, hydroxyl group from this fatty acid and hydrogen from here, and then a hydroxyl group from here and hydrogen from here. So three water molecules are eliminated and a molecule formed will have CH2 O, C double bond O, and R, CH O, C double bond O, and R, CH2 and O, P double bond O, and OH. And here this will have an OH as well. So a glycerol molecule here has two fatty acids attached here and a phosphate group attached here. This molecule can be represented showing a head which has a glycerol molecule and a phosphate group here, and two tails showing this. These tails are non-polar, so they are hydrophobic, whereas this head is polar, so this is hydrophilic. A phospholipid molecule has a hydrophilic head and two hydrophobic tails. These properties of a phospholipid molecule make it a substance to be the best substance as a component of cell surface membrane. So let's see behavior of a phospholipid in water. We take small amount of water in a beaker and place some phospholipids into it. Phospholipids would arrange themselves in such a way in this water that heads would be in the water that dissolve in the water and make hydrogen bonding with the water, whereas the tails are projected out of the water as they are hydrophobic. So hydrophobic means water hating and hydrophilic means water loving. So as this head is hydrophilic, so heads are submerged in water and make hydrogen bonding with the water, whereas the tails are projected out of the water. This makes a layer of phospholipids on the surface of water known as phospholipid monolayer. If we have large amount of phospholipids added into the water and the water has been shaken, these phospholipids would arranged in small droplets in water in such a way that their hydrophilic heads will be outwards and hydrophobic tails would be directed inwards. This makes small globular droplets known as myceles. Now look at an interesting feature which helps make you know the cell membranes with phospholipids arranged in bilayer or in two layers. For instance, if this is an animal cell has tissue fluid on outside and cytoplasm on inside. The main component of tissue fluid and the cytoplasm is water. So there is water present inside a cell and water present on outside a cell. It means uh, phospholipid molecules cannot arrange with head on one side and the tail on the other side. Why? Because both of the sides, inside and outside of this cell, has the water present. So, in this cell surface membrane, phospholipids arranged in such a way that hydrophilic heads are outwards, facing water on both surfaces, and hydrophobic tails are facing towards each other. Hydrophobic tails make hydrophobic interactions in between them, and hydrophilic heads, these are hydrophilic heads directed outwards, and these are hydrophobic tails directed inwards. So this makes a phospholipid bilayer. A cell surface membrane has phospholipids arranged in a bilayer. This could be explained on the basis of a fluid mosaic model. There are some more structures that are embedded in this phospholipid bilayer. We'll discuss the fluid mosaic model and the other substances that are embedded in the cell surface membrane. So fluid mosaic model, fluid mosaic model. Fluid here means that phospholipids make an ocean in which individual phospholipid molecules can move around within monolayer, whereas proteins are embedded in the ocean of phospholipids like islands or icebergs. So let's see a phospholipid bilayer again. So you see the hydrophilic heads are outwards and hydrophobic tails are facing inwards. Okay, so this makes a phospholipid bilayer. In this phospholipid bilayer, there are proteins embedded in it. Some proteins are embedded throughout the width of this phospholipid bilayer and are known as integral proteins. Some proteins are embedded on inside of it, called internal proteins, and some proteins may be embedded on outside of it, known as external proteins. Okay. These proteins may have carbohydrate chains attached with them, like a branched carbohydrate chain may be attached with this protein. This branched carbohydrate chain is known as glycocalyx, and this whole structure with this protein is known as glycoprotein. This glycocalyx may also be attached with phospholipids, and so this whole structure is known as glycolipid. So this is glycolipid. I'll number this one, and this whole structure is a glycoprotein. So glycolipid and glycoproteins. Other important chemical substance embedded in this phospholipid bilayer may be cholesterol molecules. As cholesterol molecules are hydrophobic, so they make interaction with the hydrophobic tails, and that are embedded in the phospholipid monolayer. So this is a cholesterol molecule. Each chemical component in the cell surface membrane or in the cell membranes have an important role to play. We'll discuss the role of each component of a cell membrane one by one. So the most abundant and most important component is phospholipid. Phospholipids. Phospholipids, as I said, they make a bilayer in such a way that hydrophilic heads are outwards and hydrophobic tails are facing each other. Okay. These hydrophobic tails provide hydrophobic barrier to polar, charged, or water soluble substances. This makes a cell membrane a partially permeable membrane. It allows some substances to pass through, and it does not allow the other substances to pass through. For instance, sodium ions, they are charged ions, so they cannot pass through this phospholipid bilayer. Similarly, glucose is a water soluble substance, so it can also not pass through this cell surface membrane or this phospholipid bilayer. However, gases like oxygen, carbon dioxide, and other gases, they can easily diffuse through the phospholipid bilayer. So this allows some substances to pass through the phospholipid bilayer, and some substances do not pass through the phospholipid bilayer. That is why it is selective in its function and is known as partially permeable membrane. The other component are glycolipids and glycoproteins. As I said earlier, proteins may have a glycocalyx attached with them on their outer side, making glycoproteins, or phospholipid molecules can have branched carbohydrate attached with them known as glycolipids. These two chemical substances present in the cell surface membrane act as either antigens, which help cell to cell recognition, or act as receptors for hormones or chemical signals. The third component is cholesterol. Cholesterol is a non-polar substance and it makes hydrophobic interactions with the hydrophobic tails. It regulates fluidity of the cell membrane. We should know what does fluidity means. Fluidity means the ability to flow. A cell membrane has viscosity of an olive oil. Cholesterol can alter the fluidity. If more cholesterol is embedded in the cell surface or cell membranes, more fluid a membrane becomes, and vice versa. Other factors which can alter fluidity of a cell membrane include number 1, temperature. Number 2, proportion of unsaturated fatty acids to saturated fatty acids. And number 3, length of the fatty acid tails of the phospholipids. And number 4, as we already discussed, cholesterol. So think about how temperature affects the fluidity. If more is the temperature of the surrounding, how would it affect the fluidity of a cell membrane? And proportion of unsaturated to saturated fatty acids. Also, how the length of fatty acid tails of phospholipids that affect the fluidity of a cell surface membrane. Think upon it and we'll discuss in questions.